This is one of a series of videos on the biology of the Florida harvester ant Pergona myrmex badius. Three decades of research, first by McKay and Porter, and more recently by Quapich and myself, have revealed the remarkable regularity and complexity of harvester ant nests and the ants and seeds within them. My casts of harvester ant nests have revealed a clear vertical structure characterized by changes in chamber shape, size, and spacing. Young workers and brood predominate deep in the nest, and older workers near the top. Seeds are stored in mid-depth chambers. Moreover, only the oldest workers leave the nest to forage and are never found below 15 centimeters deep in the nest. Middle-aged workers shuttle seeds and food between the top chambers where foragers drop them, and the brood and young workers a meter or two below. Summarized here, this suggests that workers have information about the depth at which they find themselves, and that they choose that depth actively. That this is the case can be shown in an experiment in the ant hotel, constructed of two concentric tubes perforated by holes with screen chambers attached to them. The inner tube can be rotated so the holes either gave the chambers access to the central tube or not. An equal number of old and young workers, along with some brood, were added to each chamber, and the apparatus was buried with only the top exposed. After a day of acclimation, the chambers were given access to the central tube, allowing workers to assort themselves at will. Old workers moved upward in the hotel and formed a larger proportion of the workers in the upper chambers, while younger workers predominated in the lower ones. Workers thus must have information about the depth where they are and where they should be. All of this they do in complete darkness. The nest architecture itself also implies that workers have depth information and excavate according to that depth, giving the nest its vertical organization. This nest architecture is not the result of the gradual enlargement of a small nest, for it is created in 4 to 6 days during colony relocation. The link to my video on relocation can be found on the end screen. But what might be the source of that depth information? The logarithmic distribution of chamber area suggested a template created by gas diffusion. In soils, the most likely gas gradients would be oxygen and carbon dioxide, because these would be used and produced in soil biological processes. And indeed, the distribution of chamber area looked like the mirror image of the carbon dioxide concentration. So if young workers chose high CO2 concentrations and dug very little, and old workers chose low CO2 and dug a lot, the nest would have the observed shape. And actually, young workers taken from the bottom of the nest do dig less than older ones from the top. Here are the nests dug by 200 workers. But is the CO2 gradient a consistent feature in the harvester ant nests and surrounding soils? I sampled soil CO2 by burying these vials at different depths, each with a thin plastic tubing to the surface ending in a syringe needle. The sample of soil air withdrawn from each capsule was then injected into a portable CO2 meter. Soil at all locations, along with all ant nests, showed a strong concentration gradient with depth, typically a 10-fold increase from atmospheric. Thus there was strong correlation among the ant nest architecture, ant distribution, and the CO2 gradient. But correlation is not necessarily proof of causation. An experiment is required. What if we could remove the CO2 gradient and have the ants dig a nest in this gradient-free soil? We waited for colonies to just begin a move and then bored 6 2-meter deep holes around the site of the newly forming nest. The holes were vented with PVC pipe painted black to heat in the sun and thus to draw air from the bottom of the holes. This removed the gradient in about a day. Note the 2 large arrows. If CO2 concentration is a depth Q, then the nest should lack its vertical variation and the worker distribution by age should be scrambled. Neither of these outcomes occurred. Architecture did not differ from the control nests excavated in normal CO2 gradient. The distribution of young and old workers in the nest also did not differ. No one likes to give up on such an appealing and simple hypothesis. What if I hadn't removed the gradient completely or not soon enough? It was time for a more extreme test, reversal of the normal CO2 gradient so that the high concentrations occurred just below the surface and the low ones deep. Step 1 was to remove the native CO2 gradient with boreholes as before. Note the blue points. Step 2 was to generate CO2 by dripping hydrochloric acid on limestone granules. This CO2 was delivered to the nest site through a perforated tube under a plastic sheet. This procedure dramatically reversed the gradient with very high CO2 just under the surface and low deep. The brown is the mean for the site. Step 3 was to plant a captive colony in a screen-bottom cage, so they were forced to dig a nest in the reversed gradient soil. Here's the final setup. Upon excavation, the nest architecture and worker distribution were not different from normal controls. The CO2 hypothesis had been delivered a death blow. What other cues might provide the ants with depth information? Maybe distance traveled? In this experiment, ants first contacted soil through tubes at either 2 centimeters or 40 centimeter depth. They dug chambers characteristic of their depth, suggesting travel distance gave depth information. However, when the 40 centimeter tubes were arranged so that their ends opened to soil at 2 centimeter depth, in other words horizontal, rather than at 40 centimeters, the excavated chambers were similar to near-surface chambers. From these 2 experiments, we can draw the rather unsatisfying conclusion that the ants know their depth, but not because of the distance that they have walked. For the time being, the source of the ants' depth information remains a mystery. But mystery has its own charms.